everyone and welcome to Instant Biology by Dr. Neela. The topic that we are going to deal with today is Mung bean endonuclease. Now you might be knowing that I am already running a series on RDT, basically restriction digestion I have talked about, and I have also talked about the various aspects of gene cloning. So now today what I am going to talk about is a very important enzyme that is often utilized in the case of recombinant DNA technology, and that is Mung bean endonuclease. I would be talking about what is the basic structural features of Mung bean endonuclease. I would also be talking about what is the mechanism of action. And finally, I would be talking about what are the advantages of Mung bean endonuclease. So let us now start with what is Mung bean endonuclease. So I've already made a video in Hindi about this Mung bean endonuclease. This is just a repeat of the video in English so that all our students are on the same page. So Mung bean endonuclease, what is it? It is a small glycoprotein. What is it? It is basically a small glycoprotein. Now what do you mean by glycoprotein? When I say glycoprotein, you should understand that it is having two moieties. That means it is having a proteinaceous nature also, plus it is also having a carbohydrate nature also. It is having a protein nature also, and it is also having a carbohydrate nature. That is why I am calling it as glycoprotein. Now, what is the molecular weight of this Mung bean endonuclease? The molecular weight is 39 kilodalton. Basically, this Mung bean endonuclease is comprising of two subunits. That means there are two entities, two subunits that are linked together by disulfide linkages, and that both the subunits together are combinedly called as Mung bean endonuclease. Now, if I draw like this, something like this, so two subunits, if I draw something like this, that this is something like this, and again I use another color. Now this basically can be, this is just a rough estimate of what Mung bean endonuclease would look like, and there are multiple disulfide linkages over here. Let us say that there are disulfide linkages. Okay, so that is what I had written over here also. The two subunits are linked by disulfide bond. Now what are disulfide bond? You should be knowing that disulfide bond are the bond that are present between two sulfurs like this. Okay, and where are these sulfurs where are these sulfurs present, or in which amino acids these sulfurs are present? So you would say that they are present in cysteine. So the sulfurs from the cysteine, they combine together with the help of a disulfide linkage, and they would yield a Mung bean endonuclease. Okay, now let us move forward and discuss about the next point, that in order for this Mung bean endonuclease to function, you would require a cofactor. Okay, this cofactor is basically a metallic ion. Now you might be knowing that cofactors can be of different types, but for this particular Mung bean endonuclease, the cofactor that you require is Zn2+. This is the cofactor. And without the Zn2+, or without the this particular cofactor, the enzyme Mung bean endonuclease would not function. This is the concept. Now let us understand what is the optimum activity, or at what temperature and what basically ionic strength, or what pH does this work on. So this will function at low ionic strength, and it will function at pH around 5.0. That means it would appreciate, or it would function under acidic conditions, not highly acidic conditions, but yes, it will function at acidic conditions. So this is what you should be knowing about Mung bean endonuclease. Now let us move forward. This slide is completed. Now let us move forward. How does, or what is the mechanism of action of Mung bean endonuclease? Let me take a color. Okay, so Mung bean endonuclease is single stranded specific endonuclease. So what do you mean by single stranded? So when I say single stranded, it simply means that it will be cutting only one strand. It will not be cutting both strands. It will be cutting only and only one strand. So single stranded means cutting one strand. Next, degrades DNA and RNA to 5' phosphate mononucleotides. So another thing is that it can degrade both DNA as well as RNA. I mean DNA and RNA, it is equally has the capacity to degrade both of them. Degrades DNA and RNA to 5' phosphate mononucleotides. Now what do you mean by mononucleotides? That means one by one it will cut down. For example, at the first position ATP is present, then GTP is basically adenosine phosphate is present, then guanosine is present. So it will be breaking one by one and creating mononucleotides. Mono means just one at a time. Okay. And from the 5' end, it will be cutting from the 5' end, it will not be cutting from the 3' end. This is another very important feature that you should be remembering. Now, another thing is that the ratio between single and double stranded substrates is 30,000:1. Now, why have I given this data? I have given this data to emphasize upon the fact that always the single stranded DNA or RNA would be preferred over double stranded DNA or RNA when the enzyme is going to cut. So that means 30,000 single stranded DNA molecules or RNA molecules will be cut, and in that ratio only one would be cut in the double stranded orientation. Okay. Now, double stranded DNA, double stranded RNA, DNA RNA hybrids are resistant to this enzyme. What does this mean? Basically, all of these are double stranded DNA. Okay, double stranded DNA. Next, double stranded RNA. Next, DNA RNA hybrids. These are all resistant. Basically, they will not be cut. Essentially, they will not be cut by this particular enzyme. That is why I am using the word resistant over here. Okay, now what do I mean is, when you are extensively exposing this particular enzyme to double stranded molecules, so this is what I have written: extensive exposure to high concentration of the enzyme. When you are using high concentration of the enzyme and when you are exposing the double stranded nucleic acids for a longer period of time to this enzyme, what will happen? Then the cutting will take place, or then the degradation will take place, but the condition should be that the nucleic acids that you are using, the double stranded nucleic acids that you are using, should be AT rich. Only then they would be degraded. Okay, so this is the concept. I believe you would have understood about this. So this is about the basic mechanism of action of Mung bean endonuclease. Let me now move to the next point. This is a diagrammatic representation in which I want to show you how Mung bean endonuclease would be functioning. Okay, now see this. Now this is this portion over here. You can see that this portion is single stranded DNA. Over here also you can see that this is a single stranded DNA. Of course, this portion is double stranded. In the same way, you can see over here, this is also single stranded and this is also single stranded, but this is double stranded. Now I have shown you, or I have told you, that 5' phosphate mononucleotides would be formed. That means it will start cutting from the 5' region and create mononucleotides from this side also, from this side also, similarly from this side also, from this side also. It will start breaking, and what you will get at the end, in both the conditions you will get something like this. That means it will stop cutting when you would get a double stranded region. That means when there is a double stranded region at the end, that means all the overhangs have been cut from the 5' end, what will happen? Then you would get only a double-stranded fragment, and then Mung bean endonuclease will stop cutting or will stop functioning as endonuclease. This is the concept. So I believe you would have got this clear. Now in the next step, denaturation has been performed. What do you mean by denaturation? The double-stranded DNA has been broken down, and you are getting a single strand over here. Further, you go for electrophoresis. You send it for electrophoresis. However, we are not concerned with the portion up till here. We just want to talk about things up till here because I am interested in digestion through Mung bean endonuclease. I believe you would have got a clear understanding about this. Let us move on to the next point. And now coming to the final slide, basically over here I wish to talk about the applications of Mung bean endonuclease, or where would you use Mung bean endonucleases. Let us start. High resolution mapping of termini in the RNA transcript. Now you understand this, that RNA majorly is a single stranded structure. Now when you start cutting from the 5' end a particular RNA, then you would be able to understand, you would be able to map the termini, because you are starting from the 5' end, right? You will be able to identify, or you will be able to map the nucleotides that are present in the RNA. This is the first thing. Next, conversion of protrusions into the blunt end, that I have already told you, something like this. If something like this is present, then this is 5', this is 3', this is again 5', this is 3'. So the region that is present from here till here will be cleaved, and from here till here will be cleaved. And you will be left with a blunt end like this. This is the blunt end structure that you would be left with. Okay. Next, creation of new restriction sites following overhangs removal. Now the new strand that is present over here, it will comprise of new restriction sites. Definitely, previously different restriction sites would have been present. Now the new restriction sites will be present. This is what I am mentioning over here. Creation of new restriction sites following the overhang removal. When you would be removing these overhangs, these are called as overhangs. Let me just abbreviate it, overhangs. Okay, so when you are removing the overhangs, the new restriction sites will be formed. Finally, investigation of the DNA secondary structure. What happens is, at times the DNA secondary structure can form like this. Suppose I am saying that hairpin loop has formed, or something like this has formed. What you can do is, you can investigate, you can study this using the Mung bean endonuclease. Okay, why? Because it will be cleaving the single stranded structure. I hope this video was clear to you, and you would have got a clear understanding about Mung bean endonuclease structure, its function and its applications. So thank you so much. If you liked the video, please hit the like button. Do not forget to subscribe the channel and send it to your friends also. Thank you and have a good day.